Thank you so much for coming back and being here today. So we are going to talk about antibodies and antigens. Yeah? Okay. Got a lot of slides to go through today because it's very, very fundamental. Okay. So your lesson overview for today is that antibodies are circles of proteins produced in vertebrates in response to invaders called antigen. Okay, so antibodies are incredibly diverse, specific, and are the main mediators of humoral immunity against all classes of microbes in their capability to recognize foreign molecular structure. Okay, right. Sekejap. Tadi lupa. Sabar sekejap. Okay. So, your learning objective is that by the end of this section, okay, you will, where's the laser pointer? You'll be able to explain cross-reactivity, describe the structure and function of antibodies, and identify the different classes of antibodies. Okay? If you can't hear me, let me know. Right. Okay. So the earliest reference to antibodies came from Emil von Behring and Kitasato Shibasaburo, okay, in 1890. This is where they discovered the presence of neutralizing substance in the blood that could counter infections. This was in 1890s, 1890s, not 1990, 1890s, which then led to the discovery of antibodies. So they have developed this diphtheria serum. They accomplished this by transferring serum from animals immunized against diphtheria to animals suffering from it. So this serum has the potential to cure infected animals. So we're talking about infected animals now. So in 1901, Behring was awarded the Nobel Prize for his work. And then in 1900, this is where Paul Ehrlich. Okay, wait, let me write it. Pen. Okay. 1999, 100. So this is where Paul Ehrlich suggested that the side chain theory of an antibody, where he hypothesized that the side chain cell receptors bind to a particular pathogen. He was the first to propose a model of anticorps molecule in which the antimicrobial system was branched and consisted of several places where the complementary pathway would be activated. Okay, so this model agreed with the log and key hypothesis for enzymes proposed by Emil Fischer. Mana dia Fischer? Tak apalah, I just write it down, Fischer. Okay. In 1948, Astrid Fagro described the role of B cells are specifically involved in antibody generation. And then in 1957, 1958, 1957 pula, Frank Burnett and David Talmage developed the clonal selection theory. Then in 1959, here, 1959, Gerald Edelman and Rodney Porter independently published the molecular structure of antibodies, for which later they jointly awarded the Nobel Prize in 1972. In 1973, this is the Nobel Prize in 1973. The first atomic resolution structure of antibody fragment was published, followed by the inventions of monoclonal antibody in 1975. By George Kohler and Cesar Milstein. So therefore, after 1975 is the start of modern era of antibody research and discovery, right? All these are all modern. This side is all modern. All right, so next. All right. Okay. One of the main immune response is the production of proteins, which help to fight off the antigens, which we call antibodies. Okay, antibodies are all called immunoglobulin. Alamak, peluknya. Kejap ya. Alright. Let me try to use different color lah. Let's say I take a blue color. Okay. Okay, immunoglobulin. Oh, this is green. Oh my god, I'm so sorry. Alright. They are incredibly diverse. We're talking about the immunoglobulin. They are also called antibody, yeah? Incredibly diverse and specific in the ability to recognize foreign molecular structures and are the primary mediators of humoral immunity against all classes of microbes. The antibodies can be found in the blood, the gastric and mucus secretion, also in breast milk. Okay? Right. Now, before pathogens can infect cells, antibodies in these bodily fluids can bind to them and mark them for destruction by phagocytosis. Membrane-bound antibodies can be found on the surface of B lymphocytes, which function as antigen receptor. And whereas secreted antibodies in the circulation, tissues, and mucocytes neutralizes toxins, and they prevent the entry and spreading of pathogens and eliminate microbes. Right. So before we venture into antibody structure, let's take a look at the journey before its production. During fetal life, in the bone marrow stem cells, they give rise to a B cell with a different properties than the conventional B cells. B cells are self-renewing. That mean that they can produce more mature naive B cells like themselves in the peripheral lymphoid tissue. With every stage of maturation, B cells change the location, each providing the microenvironment suitable for the B cells at that life stage. So mature naive B cells leave the marrow and enter the peripheral lymphoid tissue at the follicles. Okay, at this site, it is in the body which are composed large follicles of Peyer's patches, tonsils, and appendix. Okay, so when they encounter an antigen, the B cells receive help from T cells. Okay, listen properly. The B cells will receive help from T cells, where they divide and then they undergo somatic hypermutation and selection for B cells with higher affinity receptor. Okay, now the antibody secreting plasma. Okay, let me see if I can write it down here. Antibody secreting plasma cells are short-lived. They are short-lived. Their lifespan is very short. Oh my God. Okay. They are found in the medullary cords of the lymph nodes and the red pulp of the spleen and in the bone marrow or mucosal lamina propria. Right, kejap. Short-lived, Where were they found in? In the medullary cords. Sorry. Where is medullary cords? This one is in the lymph node, LN. Okay. Number 2, they are found in the red pulp of the lymph nodes. Of the spleen. Okay, and they are also found in the bone marrow. PM is bone marrow, or mucosal lamina propria. Okay, remember that. All right. Now, memory B cells, they can be found in the marginal zone of the spleen. Okay, the capillary sinus of the lymph and in the intestinal epithelium of Peyer's patches and crypt epithelium of the tonsils. But there are few found in the blood. That is memory B cells. Tadi we were talking about what? Tadi we were talking about secreting plasma, kan? Now we're talking about memory B cells. So I'm going to erase this now, right? Erase all slide. Okay, so I'm going to write about the memory B cells. So we talk about memory B cells. So when I say M B cells, this is my own short form. Okay, they are found in the marginal zone. Here I write it here lah. One marginal zone. Kat mana marginal zone? Spleen, right? And then the capsular sinus. Where is that? At the lymph node. And also in the intestinal epithelium. Intestinal epithelium. Okay. Where exactly in the intestinal epithelium is at the various patches. Okay, and the crypt. Apa ni? Tidak, salah agent. Crypt epithelium, Crypt epithelium. YPT epithelium. Last kali, they are found. Sorry. I'm keliru pula. Right. So, number 3. Sorry. Silap saya kat sini. Apa pula tak sama macam lymph node and lymph node dah. Okay. Second one, capsular sinus. First, they are in the marginal zone of the spleen, and then capsular sinus of the lymph node. This is correct. And then they are also found B cells, memory B cells. They are found in the intestinal epithelium. Which is in the various patches. Betul. And then number 4, crypt. CRYPT. Crypt epithelium in the tonsils. And finally, some you can find in the blood. Okay? Faham? Right. Got to check. Okay. So in this slide, we begin a discussion of antibody structure. Okay, secreted antibodies and their membrane-bound receptor, they belong to the immunoglobulin family of proteins. So immunoglobulin family are proteins. Yeah, these are a large protein family that include in the B and T cell receptors. Receptors, not the B and T cells sahaja, they are at the receptors. Okay, at the addition molecules, some at the tyrosine kinases and other immune receptors are all characterized by the presence of one or more immunoglobulins domain. Yeah, this is, let me highlight this. I don't like the green color. Let me change the color to, okay, immunoglobulin here. Domains. Yeah? An antibody is made up of V or variable. Variable, and then C, constant region. Okay, V, which means variable. C, which means constant regions, and T. And the two identical heavy chains are largely peptide units that are particularly bound to each other in the Y formation. Okay, as you can see, the famous Y formation here. Y formation here. Okay, listen properly. Which are arranged by two identical light chains. These are small peptide units, as is illustrated in on the slide. The light chain, which is represented as L, okay, light chain, they are consist of polypeptides of about 22,000, yeah, 22,000 Dalton. And heavy chains consist of larger polypeptides, around 50,000 Dalton or more. Okay, so there are 5 types of immunoglobulin heavy chain in a mammal, denoted by the Greek letters alpha, delta, epsilon, gamma, and mu. Okay, ni pen aku ni. Mu. Okay, which are also called, and there are two types of light chain. Two types of light chain, which are called lambda and kappa. Okay, today class sangat boring, tapi dia sangat because it's very fundamental. So I really need for you to really concentrate. Okay, so the region that changes to various structure depending on differences in antigens, they are called variable region. Okay, and the region that has a constant structure, they are called constant region. Simple je. Yeah. V variable region, C constant region. Right. Okay. Let's move on. Now, each of the heavy and light chain, okay, they contains an amino-terminal variable, which denoted by the V region, which consists of 100 to 110 amino acids and differ from one antibody to another. No antibody are the same. Okay, no two antibody are the same. The constant C region, okay, exhibit limited variation that defines the two light chain subclasses. Some heavy chains, for example, alpha, delta, and gamma, have contained a proline-rich hinges. Okay, like so, macam tu. Yeah, the amino terminal portions correspond to the V regions, macam ni. So these are the effector functions which are mediated by the carboxyl terminal, which is this one. These are called carboxyl terminal domain. Okay. Now the epsilon and mu heavy chains, which lack a hinge region, contains an additional domain in the middle of the molecule called carbohydrate group. Okay, like that. Right? They are linked to the heavy chain. Okay. All right. Now, so with antibodies, you need to remember number one, functional region. Okay. Right. Functional region. Number two, the types of chains. All right. Number three, the constant and variable region. And number four, the glycoproteins. Now, I mentioned immunoglobulin domains. You remember? Okay. Let's discuss a little on what this domain is composed of. Right. Now, first importantly, feature of immunoglobulin is that they each consist of series of similar but not identical sequences with about 110 amino acid long. Right? Then each of these repeats corresponds to a discrete compactly folded region of protein and are known as immunoglobulin domains. Yeah? An example is the figure on this slide. This is an Ig gene. Okay? Now its light chains has 2 Ig domains and the heavy chain has 4 Ig domains. Now the second important feature is that the amino-terminal amino acid sequence of the heavy and light chains very greatly different between antibodies. So where does this variable limit to? The variability is limited to the first 110 amino acid corresponding to the first immunoglobulin domains. So the remaining domains are constant between immunoglobulin chains of the same isotype. Alright? Okay. Alright, okay. So let's get back to this picture again. The amino-terminal variable immunoglobulin domains, or the V domains, where lies the heavy and light chains, are labeled as VH and VL. Okay? Here. VH and VL. Okay? They together make up the V region of the antibody and determine its antigen binding specificity. Now, for the C domains of both heavy and light chain, they make up the C region and they are labeled as CH and CL. CH as heavy, L as light. Okay, CH and CL. Now, the constant region has multiple heavy chain C domains and they are numbered from the amino terminal N to the carboxyl terminal N. Okay, so where is the amino acid terminal N? Which is here. Start here. kat sini nombor 1 ni kan. Here. And then it ends down to the carboxyl terminal end. This is the end. Okay? Right. Next slide. So we already discussed the basic structure of Ig domains, but within the basic structure, there are differences between the V and C region, C domains. So let's look at the picture. So each V and C domain is constructed from 2 beta sheets. Okay, 2 beta sheets. Now these beta sheets are made from several strands of beta strands. The strands contain polypeptides that are arranged in extended, flat, or conformational. Tiga type: extended, okay, here, flat, or conformation. So the beta strands are sometimes shown as ribbons with an arrow. Macam ni, dan macam ni. This one is to indicate the direction of polypeptide backbone. Okay. Now you can see example on this picture. You see that these beta strands can be packed together in a side-by-side manner to stabilize by 2 or 3 backbones hydrogen bonds between adjacent strands. Now these arrangements are called beta sheets. Yeah. The Ig domains has 2 beta sheets and sandwiched together like a 2 piece of bread. So you imagine Subway, 2 pieces of bread, they are sandwiched together. I pun lapar, tapi I puasa. Called beta sandwich. Okay? Like a sandwich, the fillings are these disulfide bonds between cysteine residues from each beta sheet. Huh? Okay. Now, the structure is called immunoglobulin fold. This one. Yeah, you can see the similarities and differences between V and C domains in the bottom panel of this figure. Yeah. Ada similarities and dissimilarities. Right? Let's see if I can change it. These 2 panels, this one and this one. Right. Okay. I don't know if you have already logged into Putra Blast, but if you haven't, I give you like 3 minutes break. Okay? I puasa so penat sikit. 3 minutes break. Kejap ya. 3 minutes break. Alright. Okay, everyone dah ada ya? Okay, welcome back. Right, so okay, a fully assembled antibody molecule comprises of three equal size of globular portion that contains of two arms joined to the trunk by a flexible and stretchy polypeptide chain. These are called hinge region. Hinge region, this one. Right. So each arm of this Y-shaped structure is formed by the linkage of light chain. Okay, with the heavy amino, sorry, with the not heavy amino. Wait. Okay, each of the Y-shaped structure is formed by a linkage of light chains with the amino terminal half of a heavy chain. So, for example, VH domain, they are paired to VL domains. Okay, let me show you here. Okay, tak nampak tadi, sorry. Right, and then the CH1 or constant region 1 domains, they are the heavy constant region. Okay, they are paired to the light TL domain like this. Faham? Okay, so when the VL, they are linked to VL, VH, alamak ni tak ada L sorry lah ya, nanti betulkan tau. Okay, these are called the antigen binding site. Then when the CH1 domain, they are paired to CL domain, they are called the trunk of Y. So remember this, antigen binding site, the trunk of Y. Okay, now let's go further down. The two antigens binding sites are formed by the paired VH and VL domains at the two arms of the Y. Imagine Y, okay, ni lah Y dia. Ni lah Y, okay. Y kat sini lagi senang lah. Y kat sini lagi senang. Okay, so these are the V region. Kira lah V light ke V heavy or dia V region. Okay, kat sini antibody, kat sini antibody or antigen binds. Okay, sorry, this one is antigen binds. Okay, they will bind here. Actually here lah, kat sini. Alright. Okay, this is where the antigen will bind. Okay. Very halus je punya tulisan ni, tapi in the books, illustration are much better lah. Just giving you a simple view of the picture. Okay, now the trunk of the Y is formed the pairing of carboxyl terminal, ni kita cerita tadi, of the two heavy chains. So we have the CH3 and CH2 domains, but CH2 does not interact. Okay, they do not interact. Okay, so the CHO, the chain, they are attached to the CH2 that lies between the two heavy chains. Itu je dia, the carbohydrate. CHO is carbohydrate lah kan. So, ini carbohydrate ni. They only attached to it, tapi dia tak, they do not interact with each other. They are just attached to the body of the carboxylic heavy chain. Yeah. Right. Okay. Constant heavy chain. Right. So, an important tool in early studies of antibody structure will the use of proteolytic enzyme if you want to break up the antibody lah. The reason behind this is to review the digestive fragments. Okay, when the antibody was cut with papain, it gives 3 fragments molecule. Okay, like this. So the papain, they cut the antibody in the amino acid terminal of the disulfide bonds that links to the heavy chains, releasing the two arms as 2 identical fragments. Macam ni. Papain cut here, papain, and then dia potong kat sini dan dia lepaskan two identical. These two lah, 1, 2, heavy chain. Right, releasing the 2 arms of identical fragments. Okay, now this is the antigen binding activity and are called the FAB fragments. Okay, FAB fragments. 2 FAB fragments. Ini. Or the antigen binding fragments. The other fragment has no antigen binding activity. It is crystallized and are called the FC fragments. FC fragments. Right. Good. Now, Now, the acid fragment correspond to the constant heavy 2, number 2, and then constant heavy number 3, CH2 and CH3 domain. Huh? So when I ask you what corresponds to the FC fragments, you must answer CH2 and CH3 domains. Okay. So another enzymes which we call pepsine. So tadi papain, and then now the pepsine pula. Now, pepsine it cuts the carboxylamine site of the disulfide bonds that produces the FAB fragments. This is when the two antigen bindings of the body molecule remain linked macam ni. They cut macam ni. But as you can see, it is still linked. Alright? Okay. Here, dah habis, potong semua. Nothing here, all gone. Okay. But here we still have the link. Okay. Right. So the remaining part of the heavy chain were cut into very small fragments. Known as PFC fragments. Here, PFC fragment. Another enzyme are called ethanol. Next time when you do Western blot, banyaklah guna ethanol, which result in two heavy chains, okay, two heavy chains and two light chains. Complexes is where the antigen cross-link with an antibody at the antigen binding site. This will form an antigen-antibody complex and sometimes called an immune complexes. Okay? Immune complexes. Are you with me, Raifa? I hope you are. Alright? Alright, so next page. So what is the function of hinge region? So the hinge region between the FC and the FAB portion are the function. For example, in the IgG molecule, it allows flexibility to some degree of independent movement of the two FAB arms. So the two arms is actually moving in your body, macam ni, macam your arms if you put at Y kan, it's actually moving. It does not just stay like that, like Y, the letter Y, no. In reality, it moves, okay? If you look at the picture on the right, the two hinge regions clearly bend differently, but the angle between the V and C domains in each of the two FAB arms is also different. Okay? So this junction at the V and C domains, okay, let me show you. Okay, that junction I'm talking about is called molecular ball and socket joint. Right? Kalau bagi gambar raja dalam exam senang gila, letak di situ molecular ball socket joint. Nah, full mark. Right? Spoiler alert. Okay, now, this junction was revealed by studies of antibodies bound to small antigens known as haptens. What are haptens? They are small antigens. They bind to immunogenic protein carrier that produces anti-haptens. So haptens are specifically recognized by antibody and can stimulate the production of anti-hapten antibodies only when they are linked to an immunogenic protein carrier. Only when they are linked to protein carrier. Okay. Right. Okay. So let's we all recap we've learned so far. Okay, first we talk about, okay, sekejap, let me see if I can change this laser pointer. Okay, first we talk about number 1. Okay, what is it? There's an amino acid sequence in C-terminal regions of the H and L change which are the same. Okay, now point 2. This is point 2. What does it do? So, variable region is an amino acid sequence in the N-terminal region of the H and L chain. This one is different. These are different. So, the region provides antibody with unique specificity. Okay. Now, point number 3. Sekejap ya. Tadi kita cakap pasal point 1, point 2, and point number 3. It's a hypervariable regions. Okay, there are regions within the variable regions. greater specificity. So this is a steep review to our next slide. Okay, this one, point 2, and then point 3. Okay? All right. Okay, so what are the classes of antibodies? There are 5 classes of antibody. There are the IgM, IgG, IgA, IgE, and IgD. Or if you have any other short synonyms, it's fine. Okay, right. So these isotypes classified in accordance to their respective heavy chain constant region. The constant region, and then the distribution and the different function of the body. Okay. The type of heavy chain constant region. Okay, first I have to write it here. Boleh tak ingat? Okay, they are classified in accordance to their respective heavy chains. Heavy chains, HAP chain. Okay, one, number 2. Constant, the distribution of the constant region. And then number 3, the different function in the body. Different function. Right? Okay. The light chains are shown in lighter shades. Okay, if you can see, the lighter shades dia macam ni lah. These are light chains. The darker shades are the heavy chains. Okay. Now, light chains are shown in lighter shades. The sulfide bonds are indicated by thick black lines. If you can see a thick black lines, that is the sulfide bonds. Now, the IgG, IgA, IgD, they have heavy chains. They have 3 domains. Okay, they have heavy chains. They have 3 domains and a hinge region. Okay, IgM and IgE, they have IgM and IgE. You tengok IgM and IgE je. And IgE. Okay, this one. These 2, okay, they have 4 domains, but they don't have hinge region. Okay, these 2, sorry. Okay, these 2, no hinge. All right. IgA, IgG, A, and D, they have heavy chains. They have 3 domains, and they have hinge. Okay, right. This one is IgM and E. Okay, they have no hinge. Now IgM and IgA, what do they do? They exist in polymeric forms, which means they contain polypeptide called the J chain. Okay, they are polymeric. Okay, they are polymeric, and they have J chain. This J chain, they are linked by 2 disulfide bonds to the Fc regions in a different monomer. Nampak tak J chain kat sini? Nampak J chain? Ada 2 disulfide bond. See the 2 disulfide bond? Here. Link to Fc region in different monomers. Okay. Now, IgM is in serum form. It's always pentamer. Always pentamer. IgA can exist in monomer, dimers, trimers, and tetramers. Okay. IgA form. For one, it can exist as monomer. 2, dimers. 3, I kat sini, trimes. And number 4, tetramers. Okay, you following me? I sure hope you do. Okay, so the summary of that part is molecule consists of an antibody molecule consists of antibody of constant and variable region for both heavy and light chains. Okay, Ig molecules are made up of domains. Each domain consists of approximately 110 amino acids. Each antigen binding sites are made up of n terminal domain, and they are also of the heavy and light chains. Okay, now apa lagi? IgM and IgE has four chains, four CH domains. Okay, that is CH1 and CH4, while IgG, IgA, and IgD have three CH domains. And at this region, hinge region is missing. They are missing a hinge region. So the hypervariable region is the variable regions of both heavy and light chains. Okay, that's it. That's a summary. Okay, that's the end of the first part. Right. Let's talk about the properties of IgG and their basic structure. I'm going to give this a summary. Okay. What differentiate the different classes of antibodies are based on the physiochemical structure and immunological properties. Okay, the IgGs, which make up about 80% of all antibodies, have heavy chains that consist of one variable domains and three identical constant domains. Now, IgA and IgD also have three constant domains per heavy chains. IgM and IgE, they have four constant domains per heavy chain. Okay, which means IgA and IgD in a constant domains, they have three heavy chain. Okay, in IgM and IgE pula, in the constant domain, they have four. Okay, four domains. Alright. So the variable domains determines binding specificity. Okay, and the constant domains of the heavy chain, they determine the immunological mechanism of action of the corresponding antibody class. Let me write it down. The variable domain, B domain. Okay, what do they correspond to? Binding specificity. Jangan tidur, please jangan tidur. Okay, the C domain, what do? They determine the immunological mechanism. Mechanism of the action, mechanism of action. Corresponding to the antibody class. Yeah? Okay? Like I said earlier, each antibody, different function, different structure, and definitely different immunological mechanism of action. Okay? Right. So it is possible for two antibodies to have the same binding specificity, but be in a different class, and therefore to be involved in different function. Okay? After an adaptive defense is produced against a pathogen, typically the plasma cells will first secrete IgM into the blood. Okay? The B cells receptors or naive cells are of the IgM class, and occasionally you will find IgD class. Yeah, mostly they are IgM class. So IgM molecules make up approximately 10% of all antibodies. Okay, prior to antibody secretion, plasma cells assemble IgM molecules into pentamers, which means macam tadi lah, there are 5 individual antibodies which are linked by a joining chain called J chain. J is correspond to joining chain, as shown in table 1. So the pentamer arrangement means that these macromolecules can bind 10 identical antigens. Yeah. However, IgM molecules, they release early in the adaptive immune response, and they do not bind to antigen as stable as IgGs, which are one of the possible types of antibodies secreted in large quantities upon re-exposure to the same pathogen. Okay? Alright, so these are few slides down are extra information that I conveniently include in this lecture. Okay, so I read in the slide lah. So IgA populated the saliva, the tears, the breast milk, and mucus secretion of the gastrointestinal, respiratory, and genitourinary tracts. So collectively, these body fluids coat and protect the extensive mucosa, about 4,000 square feet in humans. Wow. So the total number of IgA molecules in this bodily secretion is greater than the number of IgG molecules in the blood serum. So a small amount of IgA is also secreted in the serum in a monomeric form. Remember, IgA has many forms? Okay, remember this. So conversely, some IgM is secreted into the body fluids of the mucosa. Like IgM, IgA molecules are secreted as polymeric structure link with a J chain. However, IgAs are secreted mostly as dimeric molecules, not pentamers. Alright? Right, okay. IgE is present in the serum as a small quantities and is best characterized in its role as allergy mediator. Itu IgE. Now IgD pula is also present in small quantities. Like IgM, the B cell receptors of the IgD class are found on the surface of nice cells. Okay, this class supports antigen recognition and maturation of B cells to plasma cells. Alright? Okay. Fantastic. So, the function of antibodies, number one, is number one, IgG provides long-term protection because it persists for months and years after the presence of the antigen that has triggered their production. Okay, now IgG protect against bacteria, virus, neutralize bacterial toxins, they trigger complement protein systems, and they bind antigen to enhance the effectiveness of phagocytosis. The main function of IgA is to bind antigen on microbes before they invade the tissue. This one will aggregate the antigen and then they keep them in the secretion. So when the secretion is expelled, the antigen is also expelled. Yeah. IgA are also the first defense for mucosal surfaces such as intestine. Okay, such as intestine. Okay. Nose and lungs. Yeah. IgM is involved in the ABO blood group antigens on the surface of RBCs. Yeah. IgM enhance ingestion of cells of phagocytosis. IgE binds to mast cells, basophils, which participate in the immune response. Some of us think that IgE's purpose is to stop parasites, but there are other studies. Okay, there are studies on that. It can stop. And then IgD is present in the surface of B cells and plays a role in the induction of antibody production. Right? Okay. Now, antibody of the mucosal system. Now, antibodies synthesized by the mucosal immune system include IgA and IgM. Okay, they activated B cells differentiate in activated B cells. They differentiate into mucosal plasma cells, which then synthesize and secrete dimeric IgA to a lesser extent. And they also secrete pentameric IgM. Secreted IgA is abundant in tears. It is abundant in saliva, breast milk, and in secretions of gastrointestinal and respiratory tract. Antibody secretion results in a local humoral response at epithelial surfaces, and then it prevents the infection of the mucosa by binding and neutralizing pathogen. So, okay, I will go to this side. Is brief only. Okay, next. Now, here is the structure of IgG. Okay, this is the most abundant Ig in the secondary responses. This immunoglobulin can cross placenta by the neonatal Fc receptor, denoted by the FcRn. FcRn. Okay, it can activate complement. It is able to bind to phagocyte via Fc receptor. Alright. Now, this slide describes the fetal acquired by maternal IgG. So, exchange of IgG occurs at the placenta via the neonatal Fc receptor. Kat sini. Right. Okay. So, next slide. So, don't worry about the placenta crossing. I just want to share with you the general structure of the four subclasses of human IgG. This differ in the number and arrangement of the interchain disulfide bonds, denoted by the thick lines linking the heavy chains. Can you see all the thick lines? See? Sini ha. Part lain. Okay? Right. Okay. Now IgM is a pendamer. It is best activator. It is the best activator of complement. Okay? Complement system lah maksudnya. Okay? IgM is the first antibody produced in neonate and the first antibody produced after the immune response. It can exist in mucosal immune system. It is present on the surface of B cells as monomer, and it has a J chain which gives the polymeric structure. As for IgA, it exists as dimer in mucosal secretion. Involves in mucosal immunity. In circulation, IgA exists as monomer. It also has J chain, which shows that it can be polymeric and is part of secretory component. So this slide describes the information of IgA in an immune response. After activation of the plasma cells, IgA produced and released in the submucosa as a dimeric form. Then the immunoglobulin travels to the epithelial cells via the poly Ig receptor. Kat sini ha. Poly Ig receptor tahu. They cannot just simply cross something. A highway has a toll, so has a road, right? Proper road. So you have to remember that the IgA is produced where? And then it travels into the epithelial cells. How? Via the poly Ig receptor. And here it takes form into the secretory components where it enters epithelial cells. In the form of vesicle. Nampak? Ni lah dia vesicle-nya. Kat sini. Okay? Vesicle. Okay, this is the vesicle. Okay, before being injected into the human lumen by enzymatic cleavage as a secretory IgA. Alright? Okay, so the role of IgE in allergic reaction. Okay, let me read this through. IgA antibodies mediate the immediate hypersensitivity, usually that is called allergic reaction, that are responsible for symptoms of hay fever, asthma, hives, and anaphylactic shock. Any kinds of allergy lah, basically. So they bind to this antibody, the IgE, they bind to Fc receptor on the membrane of the blood basophil and tissue mast cells. So basically, it involves in immune response. Okay. So cross-linkage of receptor-bound IgE molecules by antigen, at this point they are called allergen, induces degranulation of basophils and mast cells. So a variety of pharmacological active mediators present in the granules, which are then released, giving rise to allergic manifestation. Right. Now, IgD, role unknown. Present on the surface of mature B cells as a marker. That's it. Okay. So, this table summarizes the function of each of the immunoglobulin. Okay, first we see that IgG is bound, IgG, but most abundant immunoglobulin of internal body fluids in the serum, extracellular fluids. It combats the microorganism and toxin within the body tissues. So IgA, most abundant IgA in mucous secretion, protects the external surface of the body. IgM is the first class of antibody produced during an immune response. So its presence both in internal body fluids and in the secretion. IgD, not well defined, found mostly on B cell plasma membrane as a marker. And IgE increases during parasitic infection, causes symptoms of allergy. Okay? Right. Okay, summary of that story is that IgA, IgM are secreted across epithelial surface. Okay? IgA, and IgM. IgG, IgD, and IgE can be found only within the body in serum or lymph. Okay, here. And then IgA and IgM are also found in serum and lymph, but in addition, they can also be found in secretions such as mucus secretion, saliva, and tears. Those are the upper area of IgA and IgM. So the IgA and IgM also found in external secretion, which differs from the found in serum by the presence of additional component referred to as a secretory component. Okay, secretory component here. Okay, this component is required as the IgA and IgM is transported across the epithelial cells barrier. That's why they are called secretory component. All right, next slide. So, what is the interaction of antibody molecules with specific antigen? Okay, in this section, we will look at the antigen binding site of an immunoglobulin molecule in more detail. So we will discuss the different ways in which antigen can bind to antibody and how variation in the sequence of the antibody or the V domains determines the specificity for antigens. Okay, right. Okay, so the V region of any given antibody molecule is not the same. Okay, we already established that, but I know you forgot about it, so I'm remembering you again. So the V region of the any given antibody molecules is not the same, but the sequence variability is the same. Only the distribution throughout the V region is in certain segments are not the same. If you look at the figures on the slide, you can see the variability plot that shows the amino acid sequence of many different antibody V regions are being compared to. So down here, you can identify the hypervariable regions denoted by HV1, HV2, and HV3. Okay, so in the heavy chains, they are located at residue 30 to 36, 49 to 65, and 95 to 103, respectively. So in the light chains, they are located at residue 28 to 35, 49 to 59, and 92 to 103, respectively. On the figure, you can also see the region between the hypervariable region on the V domains. So this region shows less variability and are termed framework regions. Okay, they are denoted as FR1, FR2, FR3, and FR4. Okay, right. Remember we discussed about beta sheets to slice up? Well, this is the framework form. So framework region form the beta sheet that provide the structural framework of the immunoglobulin domain. Okay, if you look at the figure on the slide, the hypervariable sequence, which is this one, correspond to 3 loops or 3 beta sheets and are positioned close to each other in the folded domain at the outer edge of the beta sandwich, which is this one. Nampak? Good. Okay. Now, when the BH and BL immunoglobulin domains are paired in the antibody molecules, the 3 hypervariable loops form each domain are brought together to create a single HV site, and this is called antigen binding site or antigen combining site. Why? Because this is the place where the specificity of the antibody towards the antigen is being determined. Okay? Because of the beta sandwich, now the site has 6 hypervariable loops, and these are termed complementarity determining regions or CDRs. Okay, we have 3 here. Why are they called CDRs? Because now the surface they form is complementary to the antigen that they bind to. Okay, so there are 3 CDRs from each of the heavy and light chains denoted by the CDR1, CDR2, and CDR3. Sometimes CDRs refers to both VH and VL domains because they both contribute to antigen's binding site. Why? Because both VH and VL combine to determine the final antigen specificity. Okay? Both variable regions. Now, in a special case, there are some antigen that binds mostly to the FAB structure at the VH CDR3 region and very few binds to the other CDRs. So in this case, the immune system will generate different combination of heavy chains and light chains, V regions, which are called combinatorial diversity. Alright? Fantastic. Senyap je on. Okay. Now, Okay, antibodies binds antigen by contacts in CDRs that are complementary to the size and shape of the antigen. Okay, now why? So the surface of the antibody molecules formed by the CDRs of the heavy and light chains is the site to which the antigen binds. Okay, the amino acids of the CDRs are different in different antibodies and are and so too are the shapes and properties of the surface created by the CDRs. So, as a general principle, antibody binds ligands whose surfaces are complementary to that of the antigen binding site. Some antigens, such as proteins, can be the same size as larger than the antibody itself. So, how do they bind? The interface between antigen and antibody, it can be extended that involves all the CDRs and part of the framework regions. Look at the figure, okay? Look at it. What small antigen? Do small proteins like heptane? Well, small antigens such as heptane and short peptides bind in a pocket lying between the heavy chains and light chains, the V domains. Look at the figure A and B. See? Okay, the surface bind to the antigen needs to be concave, undulating or convex, but not flat. This is an example of an antibody binds to HIV GP120 antigen, whereby you can see here. Okay, the black arrow is showing at the protruding finger-like shape. Okay, elongated CDR3 loops can protrude like a finger into recesses in the surface of the antigen. All right. Okay. Antigens are macromolecules, usually a molecular weight greater than 10,000, such as polysaccharide or proteins. Okay, they are recognized by the immune. Okay, sorry. Now we're talking about antigen means epitopes. Okay. Patutlah I keliru. So, alright. Antigens are molecules. They usually, molecular weight of greater than 10,000 Dalton, such as polysaccharides or proteins. They are recognized by the immune system as foreign individuals, antibodies, and are not made against the entire antigen molecules, but rather to a particular group of the molecules known as the antigenic determinants or epitopes. Macam ni. Okay, number one. So there are many different antibodies can be made against the same single antigen. So each antibody reacts with a different epitope. So, macam ni, nombor dua, nampak? Okay, alright. Complex structures such as the surface of bacterial cells may have many different epitopes, and each different antibody, they will bind to only the correct epitopes. Macam ni. Tu je. Okay. Balik semula. Okay, nampak tak? Macam ni. Okay. Alright. So, antigenic determinants on immunoglobulins, they are from glycoproteins, and they themselves are very immunogenic. Okay. These epitopes on immunoglobulins are divided into isotypic, allotypic, and idiotypic. Ingat tiga ni. Apa beza dia semua nanti akan discuss. Okay, so let's recap. An antigen determines is the region where antigen makes contact with the antibody at the antigen binding site. Kat mana? Kat antigen binding site lah. Okay, so the table on slide, they will describe the different types of antibodies that recognize immunoglobulin determinants. Anti-isotype antibodies are directed against antigenic determinants that are present at the constant region of heavy or light chains class of antibody. So the allotypes, they occur heavy or light chains in multiple allelic forms at the same isotype. So in general, two antibodies with allotypic differences will vary in one of the residue of the IG chains, and these residues are named allotypic determinants. Anti-allotype antibodies are produced by an individual immunized by antibodies derived from second animal of the same species, which carries an allotype of a specific immunoglobulin genes. For example, after an immune response has occurred, these antibodies were selected and purified from the immunized individual. So anti-idiotypes are antibodies directed against the antigen binding site of particular antibody. So these type of antibodies are generated by immunizing an animal with large quantities of purified monoclonal antibody, and they bear a single antigen binding site. Okay, I have explained one by one apa tu semua. So you baca and you betul-betul hafal and faham. Okay? Alright. So I nak rest sekejap. This is the mid-class attendance. Please take the attendance. Sekejap je. Sakit perut ke pula? Mana dia? Sekejap ya. Okay, and we're back in the now. Okay, so okay. This is isothermic determinants. Okay, it's constant determinants that define each antibody class and subclasses. So the function of antibody varies depending on which heavy chain it's used. The isotopic determinants. Alright, now this one is allotypic determinants. Allotypes are allelic variation. Okay, these are, for example, in IgG, a particular class may be slightly different between individuals. So, for example, the variation in the IgE amino acid sequence. Usually macam tu lah. The amino acid sequence would be a variation in the variation, so it will distinguish the function between the two IgGs. Although you can see that these two are actually Ig1, same kan? But because of the amino acids determinants, determines their variation and also the function. So you have to note that this type of variation has no effect on antibody function. Okay, whatever the amino acid variation, the function varies lah, but it has no effect lah on the antibody function. Not to say it's not functioning, it just has no effect. It's just an antibody sequence. Okay. Alright, so this one is ideotypic determinants. So, for example, Ig1, okay, also the same, okay, but this one is against antigen A and this one is against antigen B. So these are generated by variation in amino acid sequence in the variable heavy and light region, mostly exactly in the CDRs in the region, V region. So CDRs of V region down. So variation in the antigen binding site, these are called idiotypes. Remember, idiotype goes to antigen binding site. Right? Okay. So let's recap. Okay, the sequence variation in antibody. So number one, different light chains, no significant function effect. The different heavy chains, a very significant function effect. They have isotypic variation. Okay, that's a significant. And then allelic variation between individuals, no large function effect. Maknanya the allotypic variation. And variation in antigen body site is idiotypic variation. Itu maksudnya. Okay. Alright, so epitopes can exist as linear and discontinuous epitopes. Okay, antigens epitopes can be continuous or discontinuous. Okay, continuous or linear epitopes are part of the continuous acid sequence, and this is very helpful for antibodies that are going to recognize the linear or primary structure of a native protein that they are going to bind to. For an example, a synthetic peptide in a test tube or something along those lines. Okay. Whereas discontinuous or conformational epitopes are three-dimensional form that the antibody is going to bind to a specific parts of it. Okay. Alright. So tak salah, memang putih. I just want to show you something. Okay. Alright. So epitope. Okay, as an epitope ni besar macam ni. Okay. First, sorry. It binds to antibody. Okay. Antibody. Okay. Now, the antibody, yeah, made up of what? Sugar and carbohydrate. Okay. And they're also made up of proteins. Okay. Now, epitope is where your antibody is going to bind. Okay, the two structures that we have are going to be carbohydrate and sugars and proteins. Yeah. So epitope exists as multivalent. So epitope, multivalent. So multivalent can be divided into different epitopes or many different epitopes. This is where many different parts that antibody can bind to. Okay, or multivalent can be divided into same epitopes. Many same epitopes. Okay, so epitopes also exist as linear. Huh? Epitopes also exist as linear. Okay, and where it can bind to primary or part of primary amino acid sequence. Amino acid sequence. Okay, part of. And then they have discontinuous. They can exist as discontinuous. Epitope can also exist as discontinuous epitopes. Morning face really looks like. Cara je ke? US. Itulah. Okay. They are tertiary levels. They are bind to tertiary levels of amino acid or part of amino acid binding. Okay, what is important here is that I really want to say is that there is a lot of variation between the different types of here. Let me write it down here. Wait. There are a lot a lot of variations between different types of epitope. Sorry. Of epitopes. Yeah? Okay. Okay, I really hope you understand this. Okay? Oh, the effort I'm writing it down. Okay, so let's understand how antigen interacts with antibody that is designed to destroy. Okay, antigen, as we know, are molecule that is designed to trigger an immune response upon entering the antibody, upon entering our body lah. So interaction between the antibody and its antigen can be disrupted by high salt concentration, extreme pH detergents, and high concentration of pure epitope itself. The bindings are reversible, non-covalent interactions. The forces or bonds, in these non-covalent interactions are shown on this slide. So, electrostatic interactions occur between charged amino acid side chains. Most antibody antigen, they interact with mana dia apa tak ingat. Most antibody antigen interactions, they involve at least one electrostatic interaction. So interaction also occur between electric dipoles, as in hydrogen bonds, or short-range van der Waals forces. Okay. High salt concentrations and extreme of pH will disrupt antigen-antibody binding by weakening the electrostatic interaction and hydrogen bonds. Hydrophobic interactions, they account for most of the binding energy. Okay, this kind of interaction occur when two hydrophobic surfaces came together to exclude water. The strength is proportional to the surface area that is hidden from water. So, however, there are cases where water molecules get trapped in pockets in the interface between polar amino acid residues. In this case, the water helps with the binding and specificity of the antibody. Now, antibody also have many aromatic amino acid in their antigen binding site. Hence, this is the difference between an antibody interactions with protein antigens and natural proteins. Okay, this amino acid particle participate in van der Waals and hydrophobic interaction, and sometimes hydrogen bonds and pi-cation interaction. Okay, cation pula. Sorry. Pi-cation interaction. Alamak, tak ada orang tegur pun. Senyap je korang ni. Okay. Pi-cation interaction. Ionic interaction lah. Alright. Alright. So in this section, we look at the antigen binding site of an immunoglobulin molecule in more detail. Tadi dah detail ni lagi detail. Okay. So we discuss the different ways in which antigen can bind to antibody and how variation in the sequence of the antibody, which is the B domains, determines the specificity for antigen. Okay. Right. So the antibody-mediated immune response is also known as humoral antibody. Ni dah seronok sikit, humoral antibody. So in the host defense mechanism, they are mediated by antibody present in plasma, the lymph, and tissue fluids. So it is this type of adaptive immune response for defense against extracellular microbes, microbial toxins, and foreign macromolecules. So the cells involved in AMI, what is AMI? Antibody-mediated immune response. There are B lymphocytes, the TH cells, and phagocytic cells. Hafal ni. Okay? Alright. So, the mechanism of AMI response depends on the effective response in antibody, which are mediated. Immune response completes in 3 steps. Number 1, activation of proliferation of B cells to produce antibody. Number 2, class switching of antibody. And number 3, antibody-mediated removal of antigen. Okay? Step 1. When the B cell development and maturation completes in bone marrow, they are exported to the peripheral lymphoid tissues. When they mature, when the mature B cell in periphery is first exposed to antigen, they are activated by extensive cross-linkage of antigen to the migration receptor on B cells. Okay, the cross-linking of antigen with migration generates signal 1, which leads to internalization of antigen antibody complex by endocytosis and increased expression of class 2 MHC molecules with antigenic peptides and co-stimulatory receptor B7. Okay, so then the antigen presented with MHC2 on B cell membrane is recognized by the T cell receptor of what? TH cell. Remember number 2, TH cell? Okay. Then the activated T cells begins to express CD40 ligands, CD40L. Here. Nampak tak CD40L ni? CD40 ligand. The interaction of CD40 and CD40L will provide a signal number 2. Okay. Signal number 2, along with co-stimulatory signal provided by B7 CD28 interaction, then stimulate the B cell to begin the expression of receptor for various cytokines. Okay, the binding of cytokines released by the T cells to cytokines receptor in B cell will send the signals that support the progression of B cell to DNA synthesis and for differentiation of B cell into the antibody producing plasma cells and memory cells. Right. Step 2. Class switching antibody. So, in response to CD40 engagement and cytokines, some of the progeny of activated IgM and IgD expressing B cell undergo the process of heavy chain isotype switching. Okay? This is in response to antigen. This limit antigen, the antigen looks weird, so we need to do isotype switching. So, it leads to the production of antibodies with heavy chains of different classes such as gamma, alpha, and epsilon. So isotype switching occurs in peripheral lymphoid tissues. Where is isotype switching occurs? It occurs in peripheral lymphoid tissue. Yeah? Kat sini, peripheral lymphoid tissue. CD40 signaling and cytokinin cytokine produced by T cells will play essential role in regulatory switching to heavy chain isotypes. So the major mechanism of which CD40 signals induce the isotype switching is the induction of AID. What is AID? It's an activation-induced deaminase. It is a gene downstream of CD40 activation. Yeah? Okay. Step 3, antibody-mediated removal antigen. So, here is where antigen consists of numbers of effector mechanisms. So it needs the participation of various cellular and humoral components of the immune system involving phagocytes and complements. Okay, why? Because it involves neutralization, it involves antibody-mediated opsonization and phagocytosis, it involves antibody-dependent cell-mediated cytotoxicity or ADCC, and finally it involves the complement system. Okay? Right. So, 1. Neutralization of microbes and microbial toxin. Okay, the first step for removal of antigen is neutralization of microbes and toxin. Okay, neutralize apa? Microbes and toxin lah. Okay, neutralization is the process of which antibodies against the microbes and microbial toxins, they are block the binding of these microbes and toxins to cellular receptor. What happens then? It inhibits the infection. Okay, neutralization of microbes and antibodies will occur at different ways, many ways. So microbes enter the host cells, they bind off their surface molecule to host cell cellular receptor antibodies, and then they bind to this microbial structure and interfere with the ability of microbes to interact with cellular receptors. This interaction is called steric hindrance. Okay? Remember here, steric hindrance. Okay? Very few antibodies bind to microbes and induce conformational change. Where at the surface molecules, which then prevent microbes from interacting with cellular receptor. Now, this interaction is called allosteric effects of antibodies. Ingat ni. So, microbial toxin, tunggu tak lagi ke? Oh, never mind lah. Never mind, Tony. It's okay. The forms are not concerned. Apa ni? It's okay. I tak buka kot tadi. Lupa. Anyway, microbial also cause pathogenic infection by binding a specific cellular receptor. Okay, the antitoxin, antibodies. Sekejap, aku rasa tak ada Tony hantar. Atau dah buka. Okay. Again, microbial also cause pathogenic infection by binding to specific cellular receptors. The antitoxin of the antibodies lah. Antitoxin maknanya antibody lah, the antitoxin what? They sterically hinder the interaction of toxin to cellular receptor of host cells and prevent the tissue from injury and disease. Further development of disease lah maksudnya. Right? Okay. So, the NK cells, so number 2 is antibody dependent cell mediated cytotoxicity. What is ADCC kan? So, NK cells and other leukocytes, they will bind to antibody-coated cells by the receptor and destroy the cells. This process is termed antibody-dependent cell-mediated cytotoxicity. Okay. Here the FCYR3 is present on NK cells which bind CYR ini FC gamma lah sebenarnya ataupun 3. These are present on the surface of the NK cells. They are then bind to antibody-coated cells. Okay, since the FCY gamma R3 is also a low affinity receptor. Okay, it's a receptor, low affinity receptor. It can bind only to IgG bound infected cells, but not bind circulating monomeric IgG. So therefore, ADCC takes place only when the target cell is coated with antibody molecules. Yeah? Interaction of Fc gamma receptor of NK cells is with IgG coated target cells. So it activates the NK cells to synthesize and secrete cytokines such as interferon gamma and discharge the contents of the granules. Okay, which leads to lysis of antibody coated cells. I think so lah. Betul lah. Yeah, betul. Then eosinophils, they mediate a special types of ADCC. Okay. They directed against helminthic parasites. Kan I cakap tadi IgE tu something to do with IgE pula itu. So helminths are too large to be engulfed by the phagocytes and their integuments is relatively resistant to the microbicidal products of neutrophils and macrophage. So they can be killed by a basic protein presence in the granules of eosinophils. This is where IgE comes into action lah. IgE will coat the helminths and eosinophil can then bind to IgE through the Fc gamma R1 receptor kat sini. So this binding activates eosinophil to release their granules content, which then result in killing of helminth. Okay. Alright. Few more slides, just a couple more slides, okay? Don't worry. So, alright. So number 3 is antibody-mediated opsonization and phagocytosis. Yeah. So monoclonal phagocytosis and neutrophils, they will express a variety of surface receptors, which they are directly bind microbes that ingest them and cause intracellular killing and degradation. Now, mononuclear phagocytosis and neutrophils, they will express receptors for Fc portion of the IgG antibodies, which are specifically bind antibodies coated maknanya opsonized lah by particles. A product of complement activation called the C3b, C3b can also opsonize microbes and phagocytose by binding to a leukocyte receptor for C3b lah. So the process of coating particles for phagocytosis is called opsonization. Okay, antibodies and complements proteins that perform their functions are called opsonins. Okay, those that perform, those that apa nama dia? Perform the phagocytosis are called opsonization, and the antibodies and complements protein that perform that opsonization are called that function are called opsonins. Yeah. You need to know the difference between an opsonins and opsonization. Okay? Alright, so antibodies and complement protein that perform their functions are called opsonins. Okay, so now phagocytosis of IgG, they are coated particles, is mediated by binding of the Fc portion of opsonizing antibodies to the FCYR gamma R receptor on phagocytosis. So antibodies bound to antigens from multivalent arrays are bound to phagocyte Fc receptor with much higher avidity and free antibodies. So binding of multiple FCR to antibody-coated particles, which lead to engulfment of particles and their internalization of phagocytic vesicles. And these phagosomes, they will fuse with lysosomes and the phagocytose particles, and then they are destroyed in phagolysosome. So phagocytosis is also activated by the signal transduced by binding of opsonized particles to FCYR1 receptor. Huh? The signal results in activation of tyrosine kinase in the phagocytes, and then they are stimulated by production of various microbicidal molecules. Okay, so phagocyte activation, they will lead to production of enzyme phagocyte oxidase. Okay, and which their function is to catalyze intracellular generation of reactive oxygen intermediates that are cytotoxic or phagocytose microbes. Okay, two large microbes that cannot be phagocytized are killed by the activation of leukocyte that is capable of what they call it extracellular killing by the secretion of hydrolytic enzymes and reactive oxygen intermediates. Macam itulah cara dia. Okay, apa more? Now, I promise you this is my last slide. Okay, so complement system is a system of lactic enzyme found in the blood. These lactic enzymes are usually inactive. They are activated only under condition to generate products that mediate various effector function of complement. Now, activation of complement involves a sequential proteolysis of protein to generate newly assembled enzymes complex with proteolytic activity. Okay. Another word that you need to remember is zymogens. These are termed as proteins that gain proteolytic enzymatic activity by the action of other proteases. Zymogens. So the process of sequential zymogen activated is an enzyme cascade. Proteolytic cascade also tremendous amplification because each enzyme molecules activate at one step can generate multiple activated enzyme at the next steps. Cascade lah. So the products of complement activation becomes covalently attached to microbial cells surface or to antibodies bound to microbes and to other antigens. Okay? Alright. Alhamdulillah, that is my last slide. You all boleh rehat and then balik rumah baca balik. Okay? Thank you so much. Thank you so much for pretending to hear me. Or if you're not sleepy lah. I know it's tiring, but I've got no choice. Alright. So don't worry about this mid-class attendance because Teams takes your attendance as well. Teams takes your attendance and I think PB dah buka kan? So I think that's about it. Any questions? Okay, dah stop sharing kan? Okay, any questions? Tak ada? Class finish early. 30 minutes early, less than that, 27. So thank you very much. Really take care of yourself. I'll see you next week. Yeah. Bye. Assalamualaikum. Sesi ada message ke? Oh, the form is accepted pula. Ayo. Okay. So cepat-cepat isi. Aduh, isi je lah. You're welcome everyone. Bye-bye. Take care. Selamat Hari Raya. Still Raya lagi.
Thank you, Doctor.